Hello everyone, today we will be presenting extraction of crude from Pandanus Amaryllifolius. We are from group 1.
Thank you, Vanessa. So now I'll be proceed on the introduction and also for the objective. So plants have been traditionally essential to human survival because they offer future medicine, food, and also a shelter. And they also have been utilized to enhance the health and illness treatment. However, the using of raw materials plant directly might lead to a negative effect. So extraction techniques are used to separate the active molecules in order to overcome these difficulties and also improving the safety of the treatments. So what is exactly this extraction? So extraction is the process of separating medicinally active components from plant tissues using specific solvents. The resulting product, which may be in liquid or semi-solid or powder form, are used in pharmacy formulation or external applications. So solid extraction is the most effective of the many extraction methods that a solvent is continuously cycled through the plant's material in this percolation process, which guarantees complete extraction of bioactive compounds. So we are using and we are choosing the Pandanus Amaryllifolius as the plant for this extraction process. So the objective of this experiment is to use the Soxhlet extraction method to recover the crude bioactive components from the leaf of the pandan, and also attempts to isolate the active metabolites by evaluating and contrasting the efficacy of ethanol and hexane as solvent for extracting these bioactive chemicals. Now I'll be pass this presentation to the next group member. Thank you.
Hi, Assalamualaikum warahmatullahi wabarakatuh. My name is Isa Ileni binti Martuni. My metric number is J228024. I will present from a part of this experiment, which are materials and methods. For the materials and apparatus, we use pandan leaves, ethanol, grinder, analytical balance, heating mantle, extraction thimble, solid apparatus, and also rotary evaporator. For the methods, firstly, the pandan leaves to be extracted were collected, washed thoroughly, and deterred, and other impurities from them were removed. Next, the pandan leaves were cut into small pieces and incubated in an oven overnight. On the next day, the dried pandan leaves were made into powder by using a grinder. Then, 10 grams of the powder was measured. It was packed into an extraction thimble and placed in the solid thimble. 200 ml of ethanol was added in a 500 ml round bottom glass, and the thimble upper part was connected to a condenser. The apparatus was kept on a heating mantle, and it was heated at the boiling point of the ethanol. The process of extraction was carried out for 8 hours. After 8 hours of the extraction, the extract collected was concentrated by using a rotary evaporator. Lastly, the final extract was air-dried to get fine powder. Thank you.
Hi, my name is Divyashri. My matrix number is J21A1077. So I'll be explaining about the results part. So as we can see, the extraction solvent has two. One is ethanol and one is water. So the initial weight of dry powder is 10 grams each. So the final weight of extractions for ethanol is 158.97 grams. And for water is 159.64 grams. So the texture of extracts for ethanol is darker, more viscous, concentrated aroma. Whereas for water, the texture of extract is lighter, less viscous, and milder aroma. So the water extraction resulted in a significant higher yield than ethanol extraction, highlighting its efficiency in dissolving polar compounds. So the texture of ethanol extracts were darker and more viscous due to the presence of chlorophyll and other non-polar compounds. So water extracts were lighter and less viscous, indicating of higher polar compound solubility. Thank you.
So next we have the comparison between ethanol and water extraction from fresh pandan leaves. Shows that both methods start with 50 grams of fresh leaves and yield 10 grams of dry powder. However, ethanol extraction results in a final extract weight of 1.07 gram with a total extract yield of 158.97 gram, while water extraction yields 0.36 gram with a slightly higher total extract yield of 159.64 gram.
Hi, my name is Narsha Isha, and my number matrix is J22A3020. Today I would like to present about discussion on crude extraction from Pandanus amaryllifolius, which is pandan leaf. So, Pandanus amaryllifolius, commonly known as pandan leaf, is a tropical plant renowned for its distinct aroma and flavor. This plant is widely employed in South Asian culinary practice, where the leaf are utilized to infuse rice, dessert, and drinks with a sweet floral fragrance. The unique scent is primarily due to the presence of the compound 2-acetyl and 1-pyrroline, which is characteristic of pandan and is also found in other fragrant foods such as jasmine rice. For extraction is a fundamental technique in biochemistry and food science, allowing for the separation of valuable bioactive compounds from plant materials. This process typically involves the interaction of solvent with the plant matrix to dissolve and isolate the desired compounds, which may include flavor, scents, and help promoting phytonutrients. The extraction process can vary widely depending on the nature of the target compounds and the plant material employed. The chocolate extraction technique is an efficient method for recovering bioactive compound from solid plant specimens. In this procedure, the plant material is placed in a thimble position within a specialized extraction apparatus. A solvent such as ethanol or water is heated, causing it to evaporate and subsequently condense back into the thimble where it washed the plant material. This continuous cycle enables the solvent to dissolve the compound of interest, which are then carried back into the boiling flask to repeat the process until maximum extraction is achieved. The efficiency of the chocolate extraction method is rooted in its repetitive solvent washing process, which allows for substantial penetration of the solvent into plant cells. This interaction facilitates the dissolution of various bioactive compounds, enhancing the extraction yield. The continuous circulation of the solvent not only promotes through extraction but also minimizes contact time with heat, thus reducing potential degradation of temperature-sensitive compounds. To produce 10 grams of dry pandan leaf powder for each extraction with ethanol and water, approximately 50 grams of fresh leaf, which typically contain about 80% moisture, must be used. This conversion is critical to estimating the volume of raw materials required for effective extraction. The extraction yield from the 10 grams of dry powder reveals insight into the efficiency and solubility of the extraction methods employed. After the rotavir, the mixture of pandan and ethanol is tried to remove any residual, or the mixture of pandan and water also is tried to remove any residual solvent or moisture to obtain the exact extraction. After the drying process, 8.93 grams of extraction moisture was removed, leaving 1.07 grams of pandan extraction combined with ethanol. Next, the ethanol extraction yield is 158.97 grams of extract, indicating a significant concentration of bioactive compounds. The water extraction, however, results in a total weight of 159.64 grams. This suggests that a broader range of compounds, particularly polar compounds, were effectively dissolved in water due to its inherent properties. The finding demonstrate that water extract possesses a higher yield compared to those extracted with ethanol. This discrepancy may be attributed to water's polar character, which enhance the solubility of various polar bioactive compounds such as flavonoids, tannins, and other phenolics, which are less soluble in ethanol. Consequently, estimation of active components should account for the different in solvent properties. For the term of color, texture, and odor, ethanol extract tend to be darker and more viscous, characterized by a concentrated aroma due to the extraction of chlorophyll and flavor compounds. In contrast, water extracts are generally lighter in color, with a milder aroma reflecting the solubility characteristic of compound in ethanol compared to water. To optimize extraction, consider adjusting the solvent ratio. Studies suggest that an ethanol water mixture of around 80 to 20 can improve the extraction of specific phytonutrients. Additionally, varying extraction temperature could maximize yield while protecting sensitive compounds. Extending extraction time may also enhance the dissolution of all bioactive constituents, crucial for increasing overall yields. Implementing multiple replicates will bolster reliability and reproducibility of results. Additionally, employing advanced filtration technique will ensure the removal of solid residues, improving the purity of final extract. Finally, utilizing analytical methods such as HPLC or GCMS can provide insight into the specific bioactive compound extracted, thereby enhancing understanding of their potential application and benefits. Additional insight on extraction efficiency. The comparison between ethanol and water extraction yield from Pandanus Amaryllifolius indicates that water is more effective at extracting certain bioactive compounds. Ethanol, while a good solvent for many phenolic compounds, may not extract polar compounds as efficiently as water. This difference arises because water has a distinct ability to solve a broader range of polar compounds, thereby contributing to a higher yield in the extraction process. Furthermore, the interaction of water with the cell wall components of plant material might enhance the release of bioactive compound by breaking down structural barriers more effectively than ethanol. Therefore, the lower yield observed in ethanol extraction can be attributed to solubility and solvent interaction factors, in underscoring the importance of solvent choice in extraction strategies.
Question: what is the function of solvent in this experiment? Ethanol is a polar solvent miscible with water and highly effective for extracting polar secondary metabolites. It offers several advantages, including self-preservation at concentration above 20%, low toxicity at lower concentrations, and energy efficiency as it requires minimal heat for concentrating extract. In the chocolate extraction process, ethanol facilitates efficient extraction by continuously washing the plant material, ensuring true recovery of desired metabolites without excessive solvent use. Explain the process involved in chocolate extraction. The chocolate extraction process, also known as continuous hot extraction, uses a chocolate extractor made of glass, consisting of a round bottom glass, extraction chamber, siphon tube, and condenser. Finally, powdered plant material is placed in a porous thimble within the extraction chamber, and a chosen solvent is poured into the bottom flask. The solvent is heated, evaporated, condenses, and flows back into the extraction chamber to extract the desired compound. When the solvent level in the chamber reaches the siphon top, it cycles back to the flask. This process repeats until no residue remains. What is the function of rotary evaporator? The rotary evaporator is used to efficiently remove ethanol from the plant extract under reduced pressure. allowing evaporation at lower temperatures to protect heat-sensitive compound. This process concentrates the extract into a liquid form while enabling the recovery of the solvent for reuse, minimizing waste. Conclusion: the experiment successfully utilized the Soxhlet extraction method to recover crude bioactive components. By employing ethanol as the solvent, the process demonstrates the efficiency of selective solvent in extracting active metabolites. The extraction yield concentration plant extract, which would write weight and compare with reference solvent. This study highlights the importance of solvent choice in optimizing the recovery of bioactive compound.